Our next speaker will be José Luis Lanciego, and he is professor at the University of Navarra. He is interested in the pathophysiology of Parkinson's and Alzheimer's diseases, focusing non-human primate models of these diseases. He was trained, he did his PhD in Navarra, in the University of Navarra, and then he went to Amsterdam at the University of Brugge. And today he's going to talk about gene therapy with glucocerebrosidase as a disease-modifying treatment for Parkinson's disease and related synucleinopathies. So whenever you're ready.
Okay, so good morning, everyone. Thanks a lot for coming. It's really a pleasure to be here today, speaking in front of so many good friends. So in the next 20 minutes, I will be providing you with some overview of what's going on in the lab in the last couple of years. So we get engaged in a number of gene therapy approaches with focus on glucocerebrosidase as a potential disease-modifying treatment for Parkinson's disease and related synucleinopathies. So for those of you that may be not too much familiar with the topic, I will get started by providing you with some background. Then I will move on the results we have obtained so far that remain unpublished. And then I will try to end up with a number of conclusions together with several open questions, because this is quite a new field, so there are a lot of unknowns that need to be addressed in the near future. So this is just an overview of what we call lysosomal storage disorders. So as you can see, it's quite a heterogeneous group of disorders that, altogether they can be broadly categorized into two main groups. So those in red are lysosomal storage disorders with an impact on the central nervous system, whereas those in blue are lysosomal storage disorders without engagement of the central nervous system or with a minimal one. So from now on, we will get focused on Gaucher disease. So, of course, it's a rare disorder, but it's the most prevalent one within the category of lysosomal storage disorders. It's caused by a homozygous mutation in the GBA1 gene. It's a gene that encodes for a lysosomal enzyme known as glucocerebrosidase. So in order to suffer from the disease, you need to be homozygous. But I would like to draw your attention to the ratio of heterozygous carriers. So guys with just one single copy of the mutated gene, the ratio is estimated to just one individual in every 200 normal guys in the overall population. So the incidence of heterozygosis here is quite prevalent. And this became quite popular because it has been established a direct link between GBA1 mutations and increases incidence of a number of synucleinopathies such as Parkinson's and dementia with Lewy bodies. Even the first paper appears in 1934. It was just a case report, but of course remained neglected. And then in 1996, there's a paper that reports about 6 cases of Gaucher that were all suffering from Parkinson's disease, with typical symptoms such as resting tremor, rigidity, and bradykinesia. But the final evidence, the final conclusive evidence, was provided by Eileen Sintrasky and colleagues from the NIH. So Eileen has conducted a genetic screening study of several thousands of patients in different US hospitals. And what she found is that GBA1 mutations correlates quite very nicely with the increased incidence of Parkinson's disease. And the correlation with dementia with Lewy bodies is even stronger than for PD. And this completely changed the field, because for the first time, there is a gene that has been clearly identified that a mutation of this gene promotes the appearance of a synucleinopathy. So what about the clinical phenotype? So a Parkinsonian patient with a mutation looks more or less the same as a patient with a sporadic PD. So they both respond well to the medication, although as Jordi Claremont suggested, there has been established a slightly early disease onset on 5 years on average. But what is clear is that patients with mutations are more frequently affected by neuropsychiatric symptoms and they have a faster progression to cognitive decline. And for dementia with Lewy bodies, it's more or less the same history. So the message here is that if you have a mutation, you will probably be facing a more aggressive form of the disease. So there are some data, up to 21% of brain cases that are stored in a US brain bank are positive for GBA1 mutations. Up to 17% of patients that underwent deep brain stimulation hold a GBA1 mutation. So, we can broadly consider up to 10% of patients suffering from Parkinson are going to be GBA1 mutation carriers. Although there are a substantial number of carriers that will never be converted to Parkinson's disease, and it has been estimated that roughly 30% of these guys will develop the disease by the age of 80 years. So far, this is how it works, or to be honest, how we think it works, because we don't know, we are not too much sure of which is the overall system. It has been postulated that glucose peroxidase and alpha-synuclein are forming together a vicious circle. So if you have Gaucher disease, or if you are an heterozygous carrier, the enzyme is about to lose of activity because of the mutation in the lysosome. Such a loss of activity is pretty high. You can have a residual activity of the enzyme probably below 20%. So such a loss of activity triggers aggregation of alpha-synuclein through some unknown mechanism. We don't know too much about what's going on here. We know that loss of function promotes aggregation, but we don't know through which mechanisms. And at the end, if you have alpha-synuclein aggregated, you will end up with Parkinson's disease. So this pathway accounts for 10% of patients that are GBA1 mutation carriers. But it seems this also works the other way around. So if you have Parkinson's disease, or you have alpha-synuclein aggregates in the phonolewy bodies, aggregation of alpha-synuclein per se, I mean, in the absence of the mutation, is capable of triggering loss of function of the enzyme. So this accounts for the remaining 90% of Parkinsonian patients. So the message is that if you are thinking about using glucose peroxidase as a therapy, both types of patients, no matter they have or they do not have the mutation, are potential candidates for such a therapy. So just to briefly summarize, we know that there is a direct genetic link between mutations and synucleinopathies. However, how GCase and alpha-synuclein they talk to each other remain largely unknown so far. So with this idea in mind, we get started by using AAVs to enhance GCase activity. So the overall idea is to take advantage of AAVs to enhance GCase activity in neurons in an attempt to produce alpha-synuclein clearance. So for this purpose, we have prepared two different kinds of AAVs. The first one is for disease modeling purposes. This is an AAV9, an AAV serotype 9, coding for the mutated form of human alpha-synuclein under the control of a hybrid synapsin promoter. This means when constructed this way, this AAV is going to be neurospecific because, we want to start overexpressing alpha-synuclein only in neurons and not in glial cells. So the second AAV is our therapeutic product. It's an AAV coding for the GBA1 gene under the control of a constitutive promoter GUS-B. It was constructed this way because with the therapeutic product, we want to enhance GCase both in neurons as well as in glial cells. So the overall idea is, to approach neurons expressing synuclein with GCase in an attempt to see to what extent we can convert these neurons back into healthy neurons. So this is the overall idea. So this is what we did in rodents. This study is already completed, not yet published. So, what we did here is we get started with bilateral injections of the AAV coding for alpha-synuclein in both the left and right substantia nigra in these mice. So we wait for 4 weeks to allow transcription and expression of alpha-synuclein. So once the process is already ongoing, but before reaching a non-returning point, then we make a unilateral injection of the AAV coding for glucocerebrosidase. So what you have here, this is immunohistochemistry for alpha-synuclein. These are coronal sections through the mice brain at the level of the substantia nigra and the striatum. So what you have on the left side of the panels is the results of 8 weeks of alpha-synuclein overexpression. Okay, you can see alpha-synuclein positive neurons, the nigra, as well as in terminals in the striatum. What you have on the right side of the picture is the same amount of alpha-synuclein, I mean 8 weeks of alpha-synuclein expression, but for the last 4 of these 8 weeks, there is an enhanced expression of glucocerebrosidase. So I think it's quite clear that enhancing GCase activity induces an almost complete clearance of alpha-synuclein, both in the substantia nigra as well as in the striatum. So this just proves that by enhancing GCase, you can get rid of alpha-synuclein. Next question is, what's going on with the dopaminergic neurons? Are these neurons remain alive? So for this purpose, we performed TH stain, and we have counted by unbiased stereology the density of neurons. And this is what is shown in the histogram here. So the blue bar represents the normal density of TH-positive neurons in a naive control mice. The green bar represents the percentage of surviving neurons with 8 weeks of follow-up of alpha-synuclein overexpression. You can see there is really above 70% of neuronal death, where the red bar represents the percentage of surviving neurons in the side of the brain treated with GCase. As you can see, the amount of neuronal death has been minimized up to 17% only. So this proves, okay, you get rid of alpha-synuclein, this is neuroprotective for the dopaminergic neurons. And the final question is to what extent these represent any kind of physiological correlate. So for this purpose, what we did is we pushed these mice with apomorphine to see to what extent these animals are going to be asymmetric enough. So can you click on this video, please? Hey. Okay, I can do it. So as you can see, when you push, no. Okay, so once you push these mice with apomorphine, you see there is a contralateral rotational behavior. So the mice is rotated to the lesion side, or contralateral to the lesion side, or what you prefer is rotating ipsilateral to the healthy side. Okay, so there is another partner here, that is microglial cells. So what's going on at the level of the microglial cells? So as far as we understand Parkinson's disease, this is a two-step phenomenon in which neurons are going to die, because of alpha-synuclein aggregation, and then in a second step, microglial cells will become hyperactive or reactive and perpetuate and exacerbate neuronal death. Our data does not support this view. So what you have here is in blue TH-positive neurons. In red, you have microglial cells stained with IVA1, that is a pan-microglial marker. What you have in green is alpha-synuclein. So our data suggests that as soon as the neurons start to aggregate alpha-synuclein, and before these neurons die, microglial cells are about to change phenotype. And microglial cells surround entirely these alpha-synuclein expressing neurons, likely in an attempt to isolate these neurons for the surrounding. Once you get rid of alpha-synuclein in the other side of the nigra by enhancing GCase activity, as you can see, microglial cells go back to the normal phenotype. Okay, so with all this data in mind, we decided to upgrade the study to non-human primates, that as you may know is our main expertise. So we go in the same way, exactly the same way, bilateral injections of alpha-synuclein, then we wait for 1 month, and we perform unilateral injections of the GBA1 coding AAV, together with a control AAV coding for notrazine on the contralateral side. So the overall idea is to let the disease going on in a natural way in the untreated side, and to compare to what extent we can modify disease progression in the side of the brain treated with GCase. And one of the main advantages, among others, of using monkeys is that we can keep track of what's going on in vivo by using micro-PET scans. So we use, as a routine, in the hydrotetrabenazine as the radiotracer of choice. And the hydrotetrabenazine is a selective VMAT2 ligand. So it identifies uptake in the presynaptic dopaminergic axons. So the message is, what are you looking here? These are coronal sections of the monkey brain. This is the normal baseline conditions, the normal uptake of the radiotracer, the status of the dopaminergic system in the putamen and in the caudate nucleus. So once we perform the baseline scan, we go ahead with bilateral injections of alpha-synuclein in the nigra, and we wait for 1 more month. So as you can see, compared to baseline conditions, there is a progressive decline on the radiotracer uptake. So the process is already ongoing. There's already some damage going on in the substantia nigra. So 1 month after the initial insult with alpha-synuclein, we make unilateral injections of glucocerebrosidase in the left substantia nigra and a control vector in the right substantia nigra. We wait for 1 more month and we repeat the scan. So this is a follow-up of 2 months of alpha-synuclein, including in the left substantia nigra 1 month of GCase activity enhancement. As you can see, the animal starts to appear as asymmetric. So in the untreated side, the lesion is still going on, whereas in the side of the brain where we have enhanced GCase activity, the uptake of the radiotracer is much better preserved. And the same holds true with a follow-up of 3 months. And this side is still degenerating, where the other side is reasonably nicely preserved. Okay, so that's the message. So the pulmonary system, as far as we can see in the micro-PET scan, seems to be much better preserved in the side of the brain treated with GCase. So with 3 months of follow-up, we sacrifice this animal. I want to draw your attention, be cautious, because this is just 1 animal. We are about to kill a number of more animals, but so far we have the results, of just 1 animal. So this is a TH immunohistochemistry in coronal sections of the monkey brain. So this is the midline, the third ventricle, internal and external divisions of the globus pallidus, and the postcommissural putamen in the left side, and the same in the right side, and this is the substantia nigra. So I think that all of us, we shall agree that the density of TH-positive neurons in the side of the brain treated with GCase, even to the naked eye, is much higher than in the contralateral side. I think this is quite clear. And it is also reflected by the intensity or the density of TH innervation in the putamen. So if you compare the intensity of the stain of the TH here compared to here, I think that, I will appreciate that the intensity is higher here. So once again, we perform some unbiased histology in this animal. So the blue bar is the density of TH-positive neuron in a naive animal. The green one is the density with 3 months of alpha-synuclein overexpression. This is roughly a 60% of neuronal death. And this neuronal death is really reduced in the side of the brain treated with GCase. So just to conclude, we think that GCase is safe, it's efficient. Enhancing GCase is a good way to reduce alpha-synuclein burden. Such a reduction is neuroprotective, although, as I mentioned before, daytime monkeys are still too preliminary. At least in our hands, we think it can be considered as a promised disease-modifying therapy, particularly well-suited for patients in early stages of the disease. And as I mentioned at the beginning of my talk, there are a number of unknowns in the field that need to be addressed as soon as possible. Okay, if you have a mutation, you will be facing let's say, a more aggressive form of the disease. But why a substantial proportion of carriers will never convert to disease, we don't know now. Then there's another synucleinopathy called multiple system atrophy, where the pathology is primarily located in oligodendrocytes. And we don't know for sure whether it's associated or not with GBA1 mutations. There are 2 papers sustaining this association, and 2 papers sustaining just the opposite. There are a number of mutations that are unrelated to Gaucher, such as this mutation variant. It's unrelated to Gaucher, but has been found in patients with Parkinson and dementia with Lewy bodies. The important issue about this mutation is a prognostic marker of progression to cognitive decline. Well, there are some questions about mild and severe mutation. I don't want to enter into too much detail. There are a couple of ongoing clinical trials with ambroxol, that is a GK chaperone. So, GKs have been appointed as a validated target by the Michael J. Fox Foundation. So it's quite clear that it seems to be a very appealing target. The debate is currently focused on which is the best way to achieve a safe, efficient, and reproducible GK enhancement for the treatment of synucleinopathies this type. Well, just to acknowledge people that have participated, I want to stress the local group here at the University of Santiago de Compostela by Dr. José Luis Lavandeira. He's a very nice person to work with, for sure you know. We have some friends in Milano and in Zurich helping us with promoters and gene sequences. And the guys of the nuclear medicine department at Clinical University of Navarra, that is our academic hospital, they are the ones in charge of micro PET scans and in quantifying micro PET scans. And that's all for the time being. Thanks a lot for your attention, and now I will be much more than pleased in taking any questions you may have in mind. Thank you very much.
Thanks. I have a question. Have you seen a clear traffic of alpha-synuclein? You injected in the striatum, am I correct?
Sorry?
You injected alpha-synuclein into the striatum, no?
No, we make a stereotactic injection directly into the nigra, into the substantia nigra.
Okay, no, just my question was, have you seen any kind of traffic of alpha-synuclein between, from the site of the injection to other side of the brain, like Virginia Lee, or?
Well, I'm not taking this result with me here today, but in this monkey, when you overexpress mutated alpha-synuclein in the nigra, you can see fibers going through rostrally, through the medial forebrain bundle to reach the striatum and the cerebral cortex. And one thing that is nice to see in vivo is that at the level of the prefrontal cortex, there are a number of pyramidal neurons in layer 5 and 3 that became labeled with alpha-synuclein. And this likely accounts for in vivo neuron-to-neuron transmission of alpha-synuclein. You know what I mean? It has been observed in a number of other animals, but in monkeys this is quite clear. They are not just 1 or 2 neurons. There are not too many, but there are much more than just a few.
Thank you. You show correlation, interaction between alpha-synuclein and GBA. I was wondering whether you check whether different alpha-synuclein mutation can interact more or less with GBA.
The only thing
it's not a matter of...
The only thing we check so far, and this has been mainly done by José Luis Lavandeira's group, if we compare the performance of wild type versus mutated form of alpha-synuclein to achieve a dopaminergic lesion. So at least what José Luis is showing is that the mutated form of alpha-synuclein is much more aggressive than the wild type form. So when dealing with mice, we can go both ways. But when trying to upgrade the results to monkeys, we have to make sure that we are using a small amount of monkeys, so we are not allowed to use as many as mutation strains we wish to test. So that's why we get just narrowly focused with this mutation.
One question was around here.
Thank you. It was really a great talk. Did you try your virus in other models of synucleinopathies?
We have AVs almost for everything. So we have AVs that are capable of being propagated in the retrograde direction. This is called AV2 retro. We have AVs called OLIG-001 that specifically target oligodendrocytes, and it's a good model of multiple system atrophy. We have AVs that are penetrating the blood-brain barrier. It works pretty nice in mice, but apparently it doesn't work in monkeys, as far as we try. So, and we have also astrocyte-specific promoters, just to restrict expression of genes only in astrocytes and not in other cells. So it depends on what you want. You go one way or another. And we also have different types of promoters that might be constitutive or just neurospecific, or so whatever you want.
Mm-hmm. So did you use your GC overexpressors in another model of Parkinson's disease?
No, this is a project that is sponsored by Cybernet. It's an intramural Cybernet program, and by the Mineco, or the old Mineco, luckily. So the issue is that the work plan is that everything that is related to rodents, mice, particularly rats, are on the hands of José Luis, the other José Luis. And everything related to monkeys is our responsibility.
Okay. Okay. Thank you.
Thank you.